Okay, ladies and gentlemen, it's 2:05. I think it's time to get started. Happy New Year, everyone, and welcome to our first meeting of 2021, NBSTRN monthly national newborn screening pilot webinar. So I'm gonna get started. My name is Jennifer Taylor, for those of you that don't know me. Just a few housekeeping items. So this is an open forum. We welcome open discussion. You have the ability to mute and unmute yourself. So all I ask is if you're not speaking, please give courtesy to the speaker and mute yourself. If you don't feel comfortable speaking on the call, you can always add your comment or question to the chat box, and I will try to monitor that and answer those questions or state those comments. And then if you have a colleague that's not able to join the call today, please let them know that NBSTRN has a YouTube website where we post these webinars a couple months after we record them, and then we'll post them a couple weeks after this is over. So this is our agenda of today. Again, welcome. First, I'm going to see if there's anybody new that we have to the call. If you are new, you can feel free to unmute yourself and introduce yourself to everyone.
How's it going? I'm Ryan Colburn. I am an interested person in newborn screening and Pompeii disease. I've recognized a lot of these names. I've reached out to a lot of you for a project that we're doing to use a lot of this really wonderful data coming out of newborn screening to improve our understanding of that community, and just kind of here to take it all in and meet some new folks and get some perspective.
Welcome, Brian, to this call, and we're happy to have you. And everybody else that's on the call, welcome too. Glad to have you back. So the way this call is going to go, I'm going to go through and review kind of what this call is about, what it's turned into, our goals for this webinar. And then we'll have our updates. We have our standing agenda where we have our updates from our pilot teams that have active or completed pilots. And then we'll open it up to others that have updates on things that they're piloting, programs that are piloting conditions, as well as states that have recently added new disorders to their panel. We have one update from NBSTRN as far as our website. And then our next call is going to be February 4th. It's always the first Thursday of each month. So we've had these monthly calls for a while now. I believe they started back when SCID was being added to the RUSP. But over time, they've been transformed into a forum where states and programs and researchers have discussed, and clinicians have discussed pilots of conditions that have been recently added to the RUSP, as well as new candidate conditions that are being considered for the RUSP. And we've had the standing agenda for a long time. We've had attendance from multiple groups, including researchers, newborn screening programs, clinicians, and the standing agenda has included that we have had updates from funded pilot studies, mostly from NIH, and then their continuation to it being added to the RUSP and where things stand there. We've also had updates from other states that are conducting pilots, or they've recently added some conditions to the RUSP. And then we've had updates also from New Steps, APHL, our federal agencies, as well as here at NBSTRN. So last year we had a lot of invited presentations. Last year we had a presentation on the tool CLEAR that was developed at Mayo. We've had a presentation from Cure SMA on SMA screening and where that stands in the here in the US. In December, we had a very good, lively discussion about Duchenne muscular dystrophy and the pilots being conducted and the validation studies that have been conducted. So this year, what I would like to know is your feedback of how you think these meetings are used and if they're still useful, and what would you like to see in the future. So feel free to comment or add in or you can email me. I will add my email address to the chat box, or you can add your comment in the chat box. I have some ideas that I've come up with what I would like to share with you. And just let me know if you're interested, or if you have any other suggestions, please let me know those too. So what we're thinking about for 2021 is NBSTRN has a new website that we've developed, and we think there's some very useful tools that might be interesting to this group as things that you can utilize. So we're thinking maybe next month having a presentation so we can go through the website. You guys can see the new features of it, the new tools, how to use it. Maybe there's other researchers that are interested in newborn screening that you think might be interested in utilizing these things. So we're thinking that might be a possible presentation. We've had some past discussions, and then it's a new pilot study that's being funded for homocystinuria. So that might be a potential topic. And we had a lot of lively discussion about SMA in the past, and there's new treatments that are being developed for SMA, new guidance on caring for patients with SMA. So that also might be another topic that we might want to hit on in 2021. So I'll open it up for a couple minutes and see if anybody has any additions they want to add. Is there anything else that they would like to see discussed in 2021, or any adjustments to the standing agenda? Just feel free to let me know. And if you need time to think about it, that's okay. We can, you can always email me. There's one comment, it says, "Hi Jennifer, I think the NBSTRN website tour would be terrific. I recall hearing it was coming, but have not had a chance to look at it." And Joanne, you're just in time. The website has just went live. So this is the perfect time to reintroduce it to the community so people know what it's about. Do you have any other comments at this time? Okay, so I'm going to move on to the next part of the agenda where we get updates from the different pilot sites. If you guys can recall from our discussion last month in December, we started including Duchenne muscular dystrophy. So we're going to try to continue having states that are piloting this disorder to report on it. So if Nikki, I know some of the New York people had a meeting, but if Nikki's on the call, would you like to give us an update on DMD?
Sure, yes, this is Nikki Armstrong. Thanks for having us here today. Sure, let me just, do you usually screen share or just do it orally?
You can actually screen share if you want to, but you, let me stop sharing my screen. If you see if you can share your screen now.
Yeah, looks like I can. And can you see?
It's coming, and yes, we can see now.
Perfect. I'm trying to pull it up full slide, and there we go. It's just the one slide, so it should be easy. And are you seeing that?
Yes, we are.
Perfect. Okay, so an update for our pilot through October 31st. We have screened 17,671 newborns. There have been 17 babies referred, so that's about a 1 in 1,000 referral rate. We've had 3 with likely pathogenic or definitely pathogenic variants associated with either Duchenne or Becker. So we're at about a 1 in 5,890 incidence. We've also had 1 pathogenic possible sialuria and 1 baby that had a single pathogenic variant for limb-girdle muscular dystrophy. So it's not clear if that baby's a carrier or if they're affected and we just can't find the second pathogenic variant. We had one baby with confirmed Alagille syndrome, one baby with possible malignant hyperthermia or persistent elevation of CK, one with a completely separate inborn error of metabolism. And then, of course, we're continuing to collect data on the screened, borderline, and referred infants, and hopeful to use all this data to move forward with Duchenne on the rust.
Thank you, Nikki. Does anybody have any questions?
Yeah, so how are you obtaining current impression?
Oh, sure. So there are a series of follow-up surveys for the parents done at various times and depending on which category they fall into versus if it's a referred but not confirmed Duchenne versus a confirmed Duchenne. But there's a series of surveys.
That's great. And what response rate are you seeing? Is it pretty good?
That's a great question. And because I'm actually with PPMD, who is sponsoring the pilot, but I'm not at the sites, which are all in New York State. So I don't necessarily have all of that data.
Okay, thanks.
I assume, but I want to confirm the 3 likely pathogenic, variants, are they all boys?
Correct. They are all boys.
So your incidence rate seems right on. Thank you.
Yes, it does.
And Nikki, have you guys seen an increase in your borderline specimens as well?
Our borderline specimens, I think we're at, sorry, I don't have that number off the top of my head. It's been fairly consistent throughout. Mm-hmm. And the vast majority of our borderline specimens have ended up normalizing with repeat samples. I think we've only had one out of the 100 and something that has not.
Do you guys intend on evaluating in a certain period of time and then adjusting that borderline or even your referral cutoff at any point in the study?
No, at this point, we're more than a year in and things seem pretty consistent. And our plan right now is babies will be screened through October of 2021, but we're not planning to change those cutoffs within the pilot.
Okay.
Of course, it'll be a question whether it's appropriate to change them if it moves to clinical care, but within the pilot, we're not.
I have a quick question. This is Lisa from California. Do you do your molecular testing before the referral, or do you refer and then do the molecular testing?
We are referring and then doing the molecular testing.
Mm-hmm.
And that, I believe, on the past presentation, you're doing another sample collection. Is that correct?
Correct. Although we are working to transition to not necessarily need that second collection. Because of the New York State genetic testing laws, we were initially limited in what genetic testing labs we could use for Duchenne because there was particular samples. But as more labs have come on board, we are working to try to be able to do the genetic testing off the dry blood sample. And so a second sample would not be necessary.
Oh, I see. So you've been using like a whole blood for the pilot study?
For the genetic testing or saliva.
Okay, but if you can show that the dry blood spot works, then yeah, you could potentially reflex to the molecular testing even before the patient is seen in the clinic, in theory.
In theory.
Thank you.
Mm-hmm.
Do we have any other questions? Other questions for Nikki? Thank you, Nikki. This is a great update.
No problem. Thank you.
Okay, I'm going to go back and share my screen again. Okay, is everybody seeing my screen still?
Yes.
Okay, thank you. We're going to move on to the North Carolina pilot. I've been trying to capture in red notes that were notes from last December's meeting or about other disorders that we've talked about. And then the new stuff will be in blue. And if I don't get it during this meeting, I will review the notes and try to update it for the next meeting. But is anybody from North Carolina on the call?
Yeah, hi Jennifer, this is Kate Kucera. I have December numbers. I haven't run the January numbers, but I can do a little update on that. So we started screening in at the beginning of November, and we also screened babies born in October or enrolled in the study. As of December, we had 1,007 babies screened. We had 6 screen positives, which were sent off for a sequencing panel. We haven't had any positive sequencing panel, and they some of those also had a repeat CK done. So no confirmed positives yet. And for SMA, we've had over 12,000 screened. We had 2 screen positives. One was confirmed and one was false.
Are you guys still screening for SMA and
Yes, we are.
because you're screening through early check, is that correct?
Correct.
Okay. Is there
Yes.
any other update of when the state is going to do that, or?
So we're expecting to hand it over to the state in the first quarter. So we're able to screen through March if needed.
Okay.
This
Any
is
other?
I
Go
was
ahead.
just going to add, Jennifer, this is Beth Boyer. Of interest, I think, is that of the other positive screen for SMA, that baby has continued to have a lot of issues. There was neutropenia early on. The baby had a false positive SCID newborn screening result and continues to undergo genetic testing for a as yet undiagnosed condition.
Okay.
And I see that Beth has a little bit more up-to-date DMD information with one persistent elevated total CK.
So one of those specimen has has repeated elevated CKMM.
Correct, CK, total CK.
Total CK.
Mm-hmm.
Any other questions for the North Carolina team?
Oh, can you clarify when you say the state will pick up screening for SMA in the first quarter, is that first quarter of the calendar year or the fiscal year?
The calendar year, that's our understanding. Yeah.
And correct me if I'm not sure, you might have reported this on, I think it does say this. The baby that is a true positive, they are receiving treatment, Spinraza. I think that's what my note says.
Yes, that's correct.
And the positive, okay, so positive, the confirmed positive had 2 copies of SMN2.
Correct.
Is there any indication that this child will transfer over to gene therapy? Do you know in their study, or is that not known?
I am not aware, but the family was interested.
This is Kristen Kleinert. I'm with the North Carolina team for follow-up. Where the family is interested, gene therapy was their first choice, but AAV9 titers were too elevated. We may try to retest in the future to see if that goes down. They are interested in potentially switching over, but right now with North Carolina, we're not sure what the coverage looks like. It seemed like with Medicaid, they were only wanting to approve one treatment. It's not clear if they would cover trying to switch over to another one. We're still investigating that.
Okay. Thank you. Thanks, Kristen. Any other questions for the North Carolina team? Okay, I'm going to move on. I know May is on the call. May, I know you gave us a great talk last month about the validation studies of Duchenne. I'm not sure if you have any other updates on that, but do you want to give us updates on Pompeii, MPS 1, or any of the other MPS 1, SMA?
In terms of Pompeii, we did 18 months of pilot study, and based on the data, our advisory group and all actually been put in the regular panel, but unfortunately, it's the rulemaking process take too long, in my opinion, but it's still in the process. So as soon as it's done, and we'll do the routine newborn screening for Pompeii. This is the Pompeii status, and I do believe I said about our SMA, and right now we are in a little bit over a year, and I don't have a new. I'm reading your red part. We don't have a really new, and so case, and we still have a 6 case. And the rate is about 111,000. And the 6 cases, we have a 2 case with 2 copy SMA2 and 2 cases with 3 copies SMA2 and 2 cases with 4 copies SMA2 and 5 cases received gene therapy. And 1 case is the similar as North Carolina. And right now it's Speranza because the AAV9 antibody titer is too high, even, they tested twice, and they did have a plan to monitor that. If that antibody titer, goes down, they potentially will replaced by gene therapy. That's how much I have.
May, this is Stan Shortino from California.
Okay.
How do you tell the difference between the 4s and
the copy numbers?
2 copy number 4 versus 4+ or 5?
And I think very fortunately, I'm pretty convinced, but I can show you some figures, and actually our manuscript is in the preparation too. We use the droplet digital PCR, and during our assay, the validation phase, and we did a test on the 4 and 5, and didn't seem to have a trouble to tell the difference. I do believe technology matters, and droplet digital PCR in our hands. And of course, we only do screening for a year, but we did a very extensive validation, and I hope someday I have time to get a thing together, because I don't know Kathy Saboda on the call or not. And working with her, we have quite a large sample sets, and with all the different combination, and the assay that performed is, in my experience, and even better than the currently viewed as a golden standard MLPA. So I have another study with the Belgian. It's why I can comment on the MLPA. At least our experience is that droplet digital PCR, the perform is better.
Okay, thanks. Yeah, our external vendor, I believe, is using droplet PCR, but we haven't seen a 4 or 5 yet. So they have raw data, and they're willing to work with us. So I think maybe we can distinguish at that point.
Yeah, I think, yeah.
Okay,
Is there an opportunity to collaborate with other states who were successful in getting their programs live before the legislative side caught up, to figure out how to not be held up for helping these folks? Like, for example, Ohio was live well before their legislative side caught up with their Pompeii program.
I think I'm not so sure what you meant. in the lab and follow-up pieces were all in place, but in order to get a regular program there, this is to me is almost all the logistical steps they have to take. All the proof process has been done. Went through our advisory group and went through the Secretary of Health, and it's all been proved. But you're just waiting for them get the slots to get a rulemaking process. A lot of stamps have to have. Yeah, in Wisconsin, it's the rulemaking process is 18 months to 24 months. I would love to see this. And I think we need a very thorough review and approve process. When it's done, why take another 18 to 24 months?
So that's a good question.
Right, it just seems like
Sorry, go ahead, Ryan.
I was just commenting. Yeah, I completely agree. It seems like a very silly reason to hold off on improving the health, just so that some people who are at no risk of being born can make a decision about it.
Yeah, it would be so. May, I just wanted to back up for a minute, since you've done all of this laboratory work already and your lab is ready to go. Can you just explain to us now how long has it been since you started the journey of the rulemaking process, and where do you guys, what's the timeline like now?
So it's making progress, and right now it's before even, when our advisory group meeting have to go through secretary office, then go to governor's office, then start a rulemaking. At least we are in the rulemaking process. And this part is kind of beyond I can predict it. And the whole COVID-19 pandemic is not helping either, because people to emphasize the things, it's a priority really switch quite a bit. So.
But since they've started, how much time has lapsed since they've started?
I thank you. I think at least it's the last our committee meeting we talked about it. I would say perhaps a good, I want to say 8 to 10 months now.
Okay, so you guys are coming up on your year mark.
Yeah, yeah.
And to the best of your understanding, what are like the broad strokes process steps that are required to go from where you've already done all the work to get this set up to the point where your program is live?
They have very detailed, a lot of office have to go through review, and also when you get there, they may sit there for a while. It's just because, newborn screening for us, is our thing, but public health in the DHS department, they have so many other things. So I think it's not, it's just to my knowledge, it just matters of time. It's not, we know that will happen, just matters of time.
Thanks for.
That's a harder thing to argue.
Thanks for your comment, Ryan, and thank you for your answer, May. I just want to state that to Ryan that, and this is Wisconsin's experience. You asked a completely different state, their process of adding conditions is completely different. So some states have a faster process and some states don't. So it's very state dependent. I'm going to move on. Is anybody, I think I did see a few folks from Georgia on the call. Is anybody able to give an update on the disorders that the state is now screening for, as well as the new pilot studies that you guys are conducting?
Yeah, hey, this is Angela. Can you hear me okay?
Yep.
Okay, great. So I don't think I saw it. Well, I think Art is online, but I didn't see Bill, so I'll go. So there's not a lot of updates to give in terms of exciting stuff. Just to update on ALD, we are live. We're still at 1 Zellweger case. We do have 2 diagnosed cases of ALD, and we have a few kids who are in the follow-up process. So
You said
that's
you have two.
two diagnosed with XALD, and
Okay, two.
still
Okay.
that same one Zellweger kiddo.
Okay.
So that's good news. That
And
one
is
seems
that-
to be going well.
Are the ALD kids males?
I believe so. I'm sorry, I was pulling up general numbers for somebody yesterday, which is why I had them on the top of my head. I can actually look for you really fast while I'm talking, but yeah, and that was out of, I believe, 11 positive screens since we restarted. This was live screening, not the pilot. We started back in May.
Mhm.
And our two ALD kiddos, that's a boy. No, it's a boy and a girl, I think is what I'm, no, two boys.
Okay.
Yeah, two boys with ALD and then the Zellweger kiddo. So it's, those are not terrible numbers in my world. And 3 of those 11 are still pending. They're still being worked up. So we'll see what happens with those. I don't have numbers on SMA, but it continues to go really well. I'm really pleased with our screening process for that. MPS 1 in Pompeii should be starting soon. I don't know if Art has any update on that, but I don't have an exact date on that. We need to kind of touch base here after the new year. And in terms of our pilot projects, proximally recycled disorders, that's been on hold, but we are I think we are really actually quite close to restarting that. So that's good news. We actually have a meeting later today to start talking about that a little bit more and see where we're at. We think we've got some better data or some new data, and we're able to incorporate that into the CLEAR tool. So we think the CLEAR tool is going to help us decrease false positives on that and make it a more feasible pilot to at least continue and get something useful out of that. And we'll let you all know when it happens. And then homocystinuria, yeah, we're still working on getting that up and running, just kind of dealing with all the IRB stuff and, planning and all that sort of thing. Hoping to get it going soon, and maybe I'll have better news for you next month, but we haven't started that yet. And that's all I've got.
Any questions for Angela?
I have a quick question. This is Kashia from APHL.
Yeah, hey.
Hi, Angela. I just wanted to clarify. So you guys started full population screening for XALD in May 2020?
Yes, ma'am.
Okay, thank you.
Sure.
Any other questions?
I just would add, with all of the reports, it's really helpful to, and most, we often get the denominator, how many people were screened, just so that we can calculate our own incidence rates and see if it matches with what we're seeing in our own states.
Yeah, I think it's this is it's a great question, and I wonder if other states have any comments on this, because I know a lot of times it's easier for you to actually say the number of specimens screened, but the number of babies might be challenging. It might be more challenging to get that number. You can maybe approximate it, but does anybody have any comments on that as far as specimen screen versus getting the actual number of babies as their denominator?
Yeah, it's certainly, from a Georgia perspective, it's possible. It's certainly easier on my end to get number of screens tested versus number of babies. On a month-to-month basis where we have this meeting, it would be a challenge, and I will not promise anything like that.
Yeah, and that is totally understandable. That is totally understandable.
But I can't, if it's helpful to anybody, our birth rate's about 130,000 a year. So this should give you a little perspective on all of that.
Any other quick comments from other states that, examine the same data?
We tracking on the babies.
You are tracking on the babies?
When we talk about birth prevalences, we use babies. We don't use screening samples.
Okay, okay. That's good to know, May. Okay, if there's no other questions for Georgia, I'm going to open it up. Is there any other states that want to report on any of the new conditions that they've added, or maybe that they're piloting or interested in piloting in their state or in their program? Anybody have added any updates on their screening process for SMA? Any new additions to the rest in 2021 yet? To their panels in 2021?
Hi, sorry, this is Jill Chance from Nebraska. I just wanted to, we weren't able to make it onto the call last month, any of us, but we added SMA November 15th of this year. So we were really excited to be able to finally add that to our panel. So, and no cases so far.
How's the screening process going?
Very well. We've not run into any bumps in the road thus far. So not had anyone come up with any genes thus far. So we are a much smaller state than Georgia. We had about almost 25,000 births last year.
So your testing is done through programmer?
That's correct.
Okay. So when they do the testing, do they tell you SMA2 copy numbers?
Yes, they do. They would. We have not had that happen yet.
Thank you.
Any other states have any other updates or additions?
Hi, this is Kim Piper in Iowa. We started our SMA screening pilot last July 1st. And just as an update, we haven't identified any cases yet, but things are going well. We have about 37,000 births a year.
So Kim, how many babies have been screening since? About 15,000, am I right?
Yeah, actually, yeah, closer to 11 right now, I think they said.
Okay.
So, yeah. So
Anytime.
yeah, that's, we were kind of waiting. We figured it would happen over Thanksgiving or Christmas or New Year's, but we got through without identifying any babies then.
Might be coming soon.
Yeah. Yep.
Any other questions for Kim? Any other states have anything else they would like to report out? Is Nebraska or Iowa, are they currently screening for MPS 1 Pompeii disease?
Iowa is not. We are conducting a review for Pompeii and MPS 1 right now.
Nebraska, we have been screening for MPS 1 and Pompeii for 2 years.
And what about XALD? I should have added that in there too.
Yes, we screen for XALD as well. So currently we screen for all the rest recommended disorders with the addition of SMA.
Okay, thank you again for your updates. I really appreciate that. I'm going to go ahead and move on, and we have a quick announcement from NBSTRN. We have launched our new website. If you go to nbstrn.org, you will find it. Please check it out. log in, and what we're encouraging everyone to do is to become a member and sign up for the website. Once you sign up, you'll be able to access our tools and some of the resources that we have available. As I stated earlier in the call, we're looking at giving a presentation to this group next month, so people can understand the new features of the website. And that's all that I have on my end. Does anybody else have any announcements on the call? Any of our partners, any of the states want to announce any new presentations or anything going on that you want the community to know about?
Can I ask a question? Quick?
Sure.
I don't know if anybody from Utah on the call, because I recently reported the first newborn screen case for GMT. So anybody comments more on that? I guess not.
Yes, I was thinking the silence is now.
May?
Yes.
This is from APHL again. So we have a webinar coming up on the 21st, and Kim Hart, she's going to be on that webinar to discuss GAMT.
Excellent. Yeah, because I think everybody knows that this disorder, it's fitting in newborn screening almost perfectly, but just because all the population data have a long time and over more than a million babies just didn't have a case until now, and Utah announced. So it's very interesting to see, and I'm more curious about is Utah have 2 screens. And I'm curious they are identified from the first screening or second screening. So.
Okay.
Kesha, is there anything else that you want to discuss about or say about the webinar that's coming up at the end of this month?
Yes, so I did send out the announcement through Collaborate, so you can register if you look at that announcement. And it's part of a 3-part webinar series. So we're going to be announcing the other 2 webinars shortly as well, but it's mostly going to focus on the disorders that are not on the RUSP. So this first webinar is going to focus on the additional LSDs that people are screening for that are not on the RUSP or that were not accepted as part of the RUSP. And then we also have Kim, who's going to talk about GAMT. So again, please register for that webinar, and we're going to announce the other 2 webinars, which will occur in February soon.
Thanks so much for that update. Is there any other, and does anybody else have any updates before I give you 12 minutes of your time back? If not, I will just conclude the meeting and ask everybody again, please check out our website, our new website, or you can find us on pretty much any of the social media platforms that are available, NBSTRN. And our next call is scheduled for February 4th at 2 p.m. Again, if you have any questions or feedback, I've gotten some of the notes from the meeting about topics that people are interested in hearing about during these meetings. But if you have anything else that you would like to add, feel free to give me email at jtaylor@acmg.net. And if you have someone that was not able to attend today, please tell them to check out our YouTube channel. Thanks again. You guys enjoy the rest of your day.
Thank you.
Thanks, Jennifer.
Thank you.
Thanks.
Thank you.
Thank you.